Hi everyone, my name is Trevor Forsberg, and today we're doing our first session of the UF Doctor of Plant Medicine interview sessions. Today we have our first guest, Alex Gannon. Alex, if you could start by introducing yourself.
Thanks, Trevor. I'm Alex Gannon, and I'm from Stuart, Florida, and kind of born and raised near the beach. Always fishing, always near the water, always being outside. I got my undergrad in at the University of Florida in biology, and during this time, this was kind of when I was unsure of what I wanted to do. I started working at a research lab, and then from there, I found the DPM program and uh, loved it ever since. And moving forward, I have this is my last semester, and yeah, looking forward to kind of moving into the next part of life.
All right, awesome. Thank you, Alex. So we'll jump right into it. As you know, as a DPM and master's combined student like yourself, you're required to complete a thesis on your research over the years. So if you don't mind telling us a little bit about why you chose that particular field of research.
Yep. So, kind of starting back from an undergrad when I was in that research lab, I worked with salmonella and tomatoes, and that's what I kind of started to find my interest in vegetable crops. And after 2 years working there, I wanted to move into something in agriculture. And my professor told me to go explore, go find something new. And so that's when I went into the DPM program and then landed working with integrated pest management strategies with Dr. Norman Lepla. And this was something that was really interesting because it's more of a applied research. And so we were focusing on finding a cultural strategy for stink bugs and tomato. Um, we were working up in Live Oak, Florida, which is about an hour north of Gainesville. And a lot of farmers have been having issues with southern green stink bug in their fields. And we wanted to find a method where they weren't using conventional pesticides. And so we found that sorghum is a good trap crop that can pull off stink bugs. And so we wanted to see kind of the behavior of these stink bugs with the trap crop and if they worked and how we can implement this to the farmers.
Awesome, awesome. That's all very, very interesting. So I'm curious, what kind of methods in terms of collecting your data did you proceed with, and how long did it take you to complete this project?
So usually with a master's thesis, it takes about 2 years. Um, it took almost 2 years exactly, and the methods were because this was applied research. It was over the summer, so out in the heat for most of the time. So we had to be very efficient with how we collected and the timing, so everything had to be consistent. Um, when we started, it was getting out there as early as we could. And with stink bugs, one of the methods that's hard that is different from a lot of other methods that are used in research is they aggregate on sorghum or on crops. So you can't really randomize how you're going to collect your sample. So we had to hand collect our methods through the whole plot, and so that did take a long time. So I had an undergraduate help, and we'd go once a week during the summer, and it was a whole day event where we start about 8:30 in the morning up there, depending on when we got there for traffic. But we'd start on both. We'd start on the same side, and then kind of move down the plot, collecting, starting with sorghum in each of these fields, and then moving into the tomato. So this was definitely this was a pilot study to start off with, and so the methods were changed throughout each study to be more efficient. So that way we could collect better data.
Wow, it sounds like out in the summer heat for a long period of time and over 2 years. That's definitely a very extended project, but it's really great. That is awesome. So do you mind summarizing a little bit of the results that you were able to collect? I know you mentioned that you found that sorghum was one of the main crops, but you could speak a little bit more about that.
Yeah, so I'll take kind of a little step backwards and kind of explain the design of the plot. So what we did was we wanted to put 3 rows of sorghum on the uh, was east and west side of the plot, and it was certified organic. So everything had to be precise with organic. With when it's certified, you have to make sure everything is exactly, the labels are properly implemented, the types of fertilizer. If we had to use pests, we didn't, but if you had to use any pesticides, everything had to be organic or you can lose that certification. So we had to get these untreated seeds and we planted 3 rows on each side of 2 rows of tomatoes. And we divided them into 3 replications. So I think it was about 20 feet was replication A, 20 feet was replication B, and 20 feet C. And so we'd have an area with sorghum and an area without sorghum. So that way we could see if there was if there was an increase of stink bugs moving into the tomatoes without the sorghum. And that was, again, because it was a pilot study, we found we needed more room to split up our our controls, because if you have 20 feet of sorghum and then it's cut off and then 20 feet of no sorghum, that's a small. It's relatively easy for the stink bugs to travel. So that was something we were looking at with the results, but we didn't find. We found when the stink bug, we found that sorghum was able to attract stink bugs and pull them off of the tomato. I don't know. It was about between 80 and 90% that we were able to intercept from the tomatoes, which is really good. But we found, that between 10 to 20% that were still in the tomatoes were causing them to be unmarketable for their damage. So this was interesting because we found this could work, but since this was an integrated pest management strategy, you can't use just one system. So kind of the next goal was to figure out what are some other systems to implement based off of these results.
Awesome. Thank you for taking the time to explain that. I do have another question, especially as you're talking about some of the results. What do you think are the next steps in terms of integrated pest management for that particular field, or to help farmers in general?
So it's a good question. It's definitely a multi-year process. For example, since I finished that first trial in the summer of 2018, we continued to implement 2 more of the same field trials in Live Oak, except each time it was some of the issues I said before about the spacing, just kind of fixing some of those methods. So the next trial after 2018, we, instead of in one plot having the control and no control, this time we had 2 plots, so we could fully kind of split apart the control. So that was something to help get better success from the data. But then on top of that is, like I said before, how IPM is not just one, can't hit one of the topics from it. The next thing we are focusing on is the organic insecticides, because there's a lot out there that have been tested, especially in literature. And so our goal is trying to figure out which one of these organic insecticides would work best for the southern green stink bug. Why it's such an issue is because when they feed on the plant or the tomato, they have it almost looks like a needle-like mouth part. It's called the stylet. And so that feeds into the plant instead of chewing. And so that's little bit of an issue to use for organic insecticides because most of them are contact and not that work inside the plant. So we want to try and find a combination of which of these insecticides would work best to get the best efficacy. So that way we can knock down that last 10 to 20%. So that way we can help the farmers get it so they can market their fruits and they won't be, their yields won't decrease because of it. So this is on top of this actually being implemented in a greenhouse now, but it's going to be used for the last field trial from what my pilot study was doing. So we're actually going to implement kind of everything that we've learned since the first year and put it all into this final field study.
All right, so it's definitely a very extended project. Now that you've completed this research, what are you most looking forward to in your future with like your career in entomology as a whole?
So one of the things, at least that I like being outside. I like working out in the field and getting to see all these, like either the insects, the issues that are going on with the insects, or some of the farmers' needs. So I think having the personal relations in the fields, being able to work very closely with farmers, and maybe extension or maybe some sort of field scout, that would be something I think that would interest me going down the road for the degree.
Very interesting. Cool. So I guess to wrap all of this up, do you have any advice to any prospective DPM or master's students on how to be successful on research like your own?
Yeah, so one of the main things that I've learned that was kind of implemented and reminded of throughout the whole, both DPM and the thesis is consistency is key. When you start, for each trial, when we started the 2018 trial, whatever we started with, you keep it the same. You don't want to change. You don't want to change what you're doing midway through because that can cause issues down the line, especially if you want to publish your work or, after the fact, if you say, okay, this wasn't working, let's try this instead. But once you commit to something, stick to it. And also don't be afraid to ask questions. We're learning. There's a lot of stuff that we don't know. And so being able to understand it, and we're always learning. So to be a learner out there, and the more that you understand, the easier it is to kind of diagnose what's happening.
Awesome. Well, that's some very valuable information you provided today, Alex. So thank you very much again. I appreciate you zooming in today to share this to our viewers. And that's all. So I hope you stay safe during the rest of this quarantine.
Yeah, thank you. Thanks for having me.